Hello. Very good evening to the student who have joined. Please let me know if I am audible. I hope I am audible. Kindly let me know if you can hear my voice and also let me know if you can see the video and the presentation. We will wait till it's 6 p.m. Then we will start today's session. Okay. Till then, please wait. Good evening to the students who have joined. Please let me know if my voice is audible. And also please let me know if you can see the presentation and my video properly. Please let me know if everything is fine with the audio, video, and presentation. Hello, am I audible? Okay, thank you for confirming. I hope the presentation is visible. If not, please let me know. We will start our session at 6 p.m. So till then, kindly wait. Okay, thank you for confirming. We will be starting our session at 6 p.m. Till then, you can let me know if you have any doubt or query from our previous any classes. Okay, any issues, if there is any issue with any of the classes or anything, please let me know. We will be starting our session at 6 p.m. Let other students join, then we will start it. Okay. Okay, so it's almost 6 p.m. Okay, 6 p.m. now. Just let me just make the call open first. Okay, so it's 6 p.m. So let me start today's session. Meanwhile, if more students join, they will join accordingly. Okay. So I hope I am audible and the screen is visible to you. If not, please let me know. Okay. So very good evening to the students who have joined. My name is Mousumi Deb. I am a PMRF fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. I am your TA for this NPTEL SWAYAM course of Medicinal Chemistry, and today it is our week 9 live session. So before starting the session, let me clarify a few things. So there will be this kind of problem solving session every week from Mondays from 6 to 8 p.m. The sessions are live on Google Meet platform, and this is the link for the class. This link will remain same for our entire semester. So you can write it down somewhere or copy it and use the same link for joining each and every class. Okay. So if any network disruption happens from my side, do not leave the session immediately. I will try to rejoin as soon as possible. And the class is always open. So if you have any issue with your network, you can rejoin at any time. During the session, you can post your doubts, your queries through the chat box. Because I am recording the session and so that I can record the session without any problem, I have muted your microphones and cameras. But the chat box is always open. You can let me know any problem, any doubt, any query, anything you can let me know through the chat box. Okay. The chat box we will use for the question answer session also. So after showing you a question, I will wait for some time and you have to answer it through the chat box. Okay. If any doubt persists after class, you can drop an email to me or you can write your queries or your problem to the discussion forum of the NPTEL SWAYAM portal. I will answer your query via both methods. Okay. And this presentation and class recording, it will be shared to you after the session. Okay. I mean, generally, I try to upload the materials the same day. If it is not possible to do the same day, I will surely do it by tomorrow. So where to find the problem solving session materials? I mean the recording and also the presentation. So you have to go to the SWAYAM portal. So when there is an announcement page, is there medicinal chemistry on the left panel, you will find there is a weekly updates for each and every material, quizzes and everything. And at the end of this left panel, you will find a problem solving session 2026, January 2026. If you click on the problem solving session recording, you will be redirected to a page which looks like this. This page contains all the problem solving session material, all the class recording, the YouTube link for each and every class. Also, you will find there is a Google Drive link. If you click on this Google Drive link, it will take you to a particular Google Drive which contains each and every presentation of our sessions. Okay, whatever we have completed till now. So we have completed, I think, 8 classes till now. So there are 8 PPTs here for each and every class. Okay, and this Google, you can download the PPTs and you can read from here. Whatever you have missed during the session, you can read from that particular PPT. Okay. So these things you can find it on the SWAYAM portal. These two things, that the recording and the class PPT. Okay. So today it is our week 9 live session. So like every live session, how we will do it? I will show you around 20 question each. On all the questions will be MCQs, right? So each and every question, now it will have 4 different options. Okay. And after showing you a particular question, I will wait for some time. Maybe I will wait for a minute or two and then I will check the chat box. Okay. You have to let me know A, B, C or D, which of the following will be the correct answer for that particular question. Okay. Even if your answer is wrong, please, whatever you think is the correct answer, try to, let me know. Okay. So because these kind of questions, this MCQ type of questions are asked in your weekly assignments. So answering during the session will help you to understand the course better, as well as it will train you for answering the assignments. Okay. And also each and every question will have a detailed answer after that particular, after I show you a particular question and after after showing you the particular answer, I will discuss it in detail so that if the descriptive questions are asked in your final examination, you can easily answer the particular question. Okay. So these things that you have to keep in your mind. So today it is our week 9 live session. So in the last class, what we have done, so in the last class, I think we have discussed about how to optimize the drug target interaction. So we have a particular drug, okay, and then what changes are required to make in that particular drug so that it can access the target better, it can bind to the target more strongly. That we have learned in our last class. Okay. So today what we will be discussing, we will be discussing the optimizing the access to the target, how the drug can go to the target more easily or more freely. Hence, we will go to a very important topic, that is prodrugs and drug alliances, endogenous compounds, peptidomimetic and oligonucleotide as drugs. Okay, so these things that we are going to discuss in today's class. Okay, so I hope I am audible, and okay, let most when most students join, they can access the class easily. So I will now start the session by showing you the first question of today's session. Okay, so the first question of today's session, it says that which of the following statements is false regarding prodrugs? Your options are: number A, metabolic enzymes can activate prodrug. Number B, in general, prodrugs are inactive compounds. Number C, prodrugs cannot be activated by photodynamic therapy. Number D, not all prodrugs are activated by esterase enzyme. Which of the following statements is false regarding the prodrugs? Again, I will repeat option number A, metabolic enzymes can activate prodrugs. Number B, in general, prodrugs are inactive compound. Number C, prodrugs cannot be activated by photodynamic therapy. And number D, not all prodrugs are activated by esterase enzyme. Which of the following statements is false regarding the prodrugs? As I have said, I will wait for a minute or two, then I will check the chat box. Read the options very carefully. Whenever there is a true false kind of question, so students tend to make mistake in this kind of question. So be careful, read the options carefully, and then let me know. A, B, C, or D, which of the following will be the correct answer for problem number 1? Which statement is false regarding prodrugs? Your options are: number A, metabolic enzymes can activate prodrug. Number B, in general, prodrugs are inactive compound. Number C, prodrugs can not be activated by photodynamic therapy. And number D, not all prodrugs are activated by esterase enzyme. Okay, let me check the chat box. Let's see if there is any answer for problem number 1. Okay, so I have received one answer saying that number B is the correct answer for our problem number 1. Means statement number B is the false statement. Okay, let me just check the chat box now. So the correct answer, the statement which is false regarding prodrugs, it is actually number C, okay, which says that prodrugs cannot be activated by photodynamic therapy. This statement is false. Okay, now let us understand what is a prodrug, what is a purpose on, and I can see there is a, okay, there is a printing mistake here. Drugs. Okay, so now let us discuss what is prodrug. So basically, prodrugs are the compounds which are inactive in themselves but which are converted in the body to the active drug. Okay, so initially the prodrug is inactive, but inside the body they get activated by some methods or some techniques or pathways that we will be discussing in our upcoming slides. Okay, so they have been useful in tackling problems such as acid sensitivity, poor membrane permeability, drug toxicity, bad taste, and short duration of action. Usually, the metabolic enzyme is involved in converting the prodrug into the active drug. So, a good knowledge of drug metabolism and enzyme involved allows the medicinal chemist to design the suitable prodrug, which turns the drug metabolism into an advantage rather than a problem. Prodrugs have been designed to be activated by variety of metabolic enzymes. Ester prodrugs, which are hydrolyzed by esterase enzymes, are particularly common, but prodrugs have also been designed which are activated by indemethylation, decarboxylation, and hydrolysis of amide and phosphate. This statement is important, that Not all prodrugs are activated by the metabolic enzyme. However, for example, photodynamic therapy involves the usage of the external light source to activate the prodrug. So our statement number C, which says that the prodrugs cannot be activated by photodynamic therapy, this is false. Okay, most of the prodrugs are activated by the metabolic enzymes like esterase or decarboxylase or N-demethylase, but some prodrugs can be activated by the photodynamic therapy also. When designing prodrug, it is important to ensure that the prodrug is activated, effectively converted to the active drug once it has been absorbed into the blood supply. But it is also important to ensure that any groups that are cleaved from the molecule are non-toxic. Okay, so this is also very important, that to understand that it is important to ensure that any group or that are cleaved from the prodrug are non-toxic. So if for example any metabolic enzyme cleaves the ester product from the with the help of the esterase enzyme, so what will happen, this particular it will be broken to the carboxylic acid and alcohol, so there will be some kind of side products are forming. Okay, so in each and every product, whatever product is activated by the metabolic enzyme, there are some side products. Okay, so it is important to ensure that any group that are cleaved from the molecule are non-toxic. Advantages of prodrugs: an increase in water solubility by using sodium succinate ester and as in chloramphenicol succinate IV injection, an increase in lipid solubility, increased duration of action by using lipid soluble ester, or increase the oral absorption by using ester of the high polar groups or N-methylation, increased topical absorption of steroid by masking OH group as the esters or acetonides, a decrease of water solubility to increase the palatability as in chloramphenicol palmitate, decreased GI irritation as in aspirin, site specificity as in methyl dopa, increased half-life and chemical stability as in cefamandol acetate, a stable prodrug which parent cefamandol is unstable, is in the solid dosage form. Hetacillin is another prodrug which is for ampicillin. Okay, one by one we will be discussing each and every example or many examples of prodrugs, and the uniqueness of each and every prodrug we will be discussing in our upcoming class. So for problem number 1, the statement number C, which says that prodrugs cannot be activated by photodynamic therapy, it is false. Okay, other statements are correct, which says that the metabolic enzymes can activate prodrugs, this is correct. In general, prodrugs are inactive compound, it is also correct. And number D, not all prodrugs are activated by esterase enzyme, this statement is also correct. Okay, so the statement number C is false. Now I will go to the next problem. The problem number 2 says, which of the following will not be a limitation for a prodrug design? Your options are: number A, formation of a toxic product; number B, an inert carrier can be cleaved off forming toxic product; number C, prodrug cleaving off before reaching the target site; and number D, the cleaving of carrier is also site specific. Which of the following will not be a limitation of prodrug design? Number A, formation of toxic product. Number B, an inert carrier that can be cleaved off forming a toxic product. Number C, product cleaving off before reaching the target site. Number D, cleaving off the carrier is site specific. Which of the following is not a limitation for product design? Again, I will wait for a minute, then I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer for problem number 2. Read all the options carefully and then let me know in the chat box A, B, C or D, which of the following you think is the correct answer for problem number 2. Which of the following will not be a limitation for product design? Okay, let me check the chat box. Let me check if there is any answer for problem number 2. Okay, I have received answer which is saying that number A is the correct answer. Okay, let me check if number A is the correct answer or not. So, the following that will not be a limitation for product design is, it is number D. The cleaving of the carrier is also site specific. This will not be a limitation of product design. So the basic thing is that production of the toxic side effect or inert carrier which can form the toxic side effect, or the product cleaving up before reaching the target size. If you read this carefully, you will find that all these are the limitation for the product design. Okay, we do not want to get a toxic side product or a toxic product or the product cleaving up before reaching the actual target site. But the cleaving of the carrier, which is site specific, it does not be a limitation of a product design. Rather, we want that our cleaving of the carrier, it has to be site specific, right? So while product design is a powerful strategy to overcome the pharmacological barriers, it faces several significant limitations and development challenges. One is the metabolic and genetic variability, enzyme polymorphism, individual differences in enzyme expression. For example, cytochrome P450 esterases mean that product may be activated at different rates or not at all certain in certain patients, leading to inconsistent therapeutic effect or unexpected toxicity. Species differences, data from animal models often fail to translate to human because metabolic pathways and enzyme distribution vary significantly between the species. Safety and toxicity concern, toxic promoieties. The masking group or promoiety released during the activation must be non-toxic and easily excreted. If it is reactive or poorly cleared, it can cause secondary side effect. Unintended activation, products may be converted into the active drug in the non-target tissues, causing off-target toxicity. And formation of the chemical formulation and chemical stability. Chemical instability, some products, particularly phosphate derivatives, are prone to premature hydrolytic and oxidative degradation during manufacturing and storage, compromising their potency before even reach the patient. Complex synthesis, the designing of a product often requires sophisticated chemical modification that must preserve the original drug's core structure. Okay, and the core structure and pharmacophore, while adding a reversible linker. So pharmacokinetic limitations, delayed onlet because of the required extra step of application once in the body. Products naturally have a longer time to peak concentration compared to the active parent drug. Incomplete conversion. If biotransformation is insufficient, the product, the significant portion of the dose may be excreted as the inactive product, reducing the medication's overall potency. Regulatory analytic challenges, complete complex monitoring. It is more difficult to monitor the concentration of both product and the active metabolite in the blood, requiring high sensitivity and analytic technique like LCMS and or MS. Then stringent approval, regulatory bodies like FDA require comprehensive safety data on both product and release promoiety, which increase the time and cost of the clinical trial. Okay, so all these are the limitations for the prodrugs. Although there are several limitations, but prodrug is a very important therapy, and it is a very important to understand what is the to design the product so that it can overcome all the limitations that are mentioned here. Okay, so I hope it is clear what are the limitations of the product. I will now go to the next problem. So problem number 3 says identify the incorrect statement. Again, it is a correct incorrect type of question. So read the options very carefully and then answer in the chat box. Okay. Number A, ester products are used to mark the carboxylic acid functional group. Number B, enalaprilate is a prodrug of enalapril. Number C, ester products should be made moderately stable. And number D, it is hydrolyzed by esterases to an alcohol and carboxylic acid which are quickly excreted due to the increased polarity. Which of the following statement is incorrect? Again, I will repeat. Number A, ester products are used to mask the carboxylic acid functional group. Number B, enalaprilate is a prodrug of enalapril. Number C, ester products should be made moderately stable. Number D, it is hydrolyzed by esterases to an alcohol and carboxylic acid which are quickly excreted due to the increased polarity. Identify the statement which you think is incorrect. Let me wait for a minute, then I will check the chat box. Whatever you think is the correct answer, let me know A, B, C or D, whatever you think is the correct answer. Identify the statement which is incorrect. Number A, ester products are used to mask the carboxylic acid functional group. Number B, enalaprylate is a prodrug of enalapril. Number C, ester product should be made moderately stable. And number D, it is hydrolyzed by esterases to an alcohol and carboxylic acid which are quickly excreted due to the increased polarity. Let me check the chat box. Okay, I have received an answer which is saying that number D is the correct answer. Okay, let me check if number D is the statement that is incorrect. Okay, so the statement which is incorrect will be, it is actually number B, which says that enalaprylate is a prodrug for enalapril. This statement is incorrect. Okay, let us discuss it. Now, what is enalaprylate? What is enalapril? So, basically, enalapril is the prodrug. Okay, not an enalaprylate is the active drug. Okay, enalapril is the ester prodrug and enalaprylate is the active carboxylic acid drug. So, now statement number B, which says that enalaprylate is a prodrug of enalapril, this is false. Okay, but rather enalapril is a prodrug of enalaprylate. Okay, so the statement number A, which says that ester products are used to mask carboxylic acid, which is correct. Number B is the false statement. Number C, which says ester products should be made moderately stable, this is also correct, and it is hydrolyzed by esterases to an alcohol and carboxylic acids which are quickly excreted due to increased polarity. This is also correct. Okay, because alcohol and carboxylic acid both are more polar than the ester products, right? Ester drugs, right? So they will be excreted more quickly rather because of this increased polarity. So now let us understand esters as a product, and esters as a product is a very vast, the most of the, not most, but rather I will say that a large number of products that are actually esters which are activated by the esterase enzymes. Okay, so products have proved to be very useful in temporary masking an awkward functional group which is important to target binding but which hinders the drug from crossing the cell membrane of the gut wall. For example, a carboxylic acid functional group may have be an important role to play in binding a drug to its binding site via ionic and hydrogen bonding. However, the very fact that it is an ionizable group may prevent it from crossing the fatty cell membrane. The answer is to protect the acid formation as an ester. The less polar ester can cross the fatty cell membranes, and once it is in bloodstream, it is hydrolyzed back to the free acid formed by esterases in the blood. Examples of the ester products are used to aid membrane permeability include the enalapril, which is a prodrug of the antihypertensive agent enalaprylate, okay, and pivamycin, which is a penicillin prodrug. Not all esters are hydrolyzed equally efficiently, and range of the esters may need to be tried to find the best one. Okay, it is possible to make esters more susceptible to hydrolysis by introducing electron withdrawing groups to an alcohol moiety, for example, OCH2CF3 or OCH2CO2R or OCONR2 or OIR. The inductive effect of this group aids the hydrolytic mechanism by stabilizing the alkoxide leaving group. Care has to be taken, however, not to make the ester too reactive in case it becomes chemically unstable and hydrolyzed by the acid condition of the stomach or more alkaline condition of the intestine before it reaches to the blood supply. To that end, it may be necessary to make the ester more stable. An example, cyclopropanoic acid ester have been studied as a potential prodrug because the cyclopropene ring has an ability to stabilize the carbonyl group of the neighboring ester. In this respect, the acting biostar of the double bond. A conjugate double bond stabilizes a neighboring carbonyl group due to the interaction of the pipe system involved. It is proposed that the sigma bonds of cyclopropene ring are oriented correctly to allow the hyperconjugative interaction similar to stabilizing effect of the neighboring carbonyl group. The interaction proposed involve the hyperconjugation donation to the antibonding pi orbital of carbonyl group. Okay, so the cyclopropene carboxylic acid ester as the product are the bioester for this alpha beta unsaturated ester. So the example now of this particular ester products, one is the enalapril, which I have said it is a ester, which is the structure of enalapril is this one, where R is the ET, and the active form is the enalaprylate, where R is the hydrogen. Okay, we also have this example candroxylate, which is for which the prodrug is this candoxtril, which has this 5-indenyl group as the as the masking group of the carboxylic acid, which is also a ester. Okay, So this is all about the ester as a product, why it is important and how to design that particular ester products. Okay. Now I will go to the next problem. So problem number 4 says which functional group ensures the efficient conversion of 6-mercaptopurine from azathioprine? Azathioprine. Your options are number A nitro, number B carboxylic acid, number C amine, and number D bromine. Which functional group ensures the efficient conversion of 6-mercaptopurine from azathioprine? Which functional group ensures efficient conversion of 6-mercaptopurine from azathioprine? Nitro, COOH, NH2 or Br? Let me know A, B, C or D which of the following will be the correct answer. Which functional group ensures the efficient conversion of 6-mercaptopurine from azathioprine? Okay, let me check the chat box. Let's see if there is any answer for problem number 4. Okay, I have received two answers. One is saying number A and another is saying that number C is the correct answer. Okay, let's find out which of the following is the correct answer for problem number 4. So the group which ensures the efficient conversion of the 6-mercaptopurine from azathioprine will be, it is number A, nitro. Okay. So sometimes prodrugs are designed to be converted slowly to the active drug, thus prolonging the drug's activity. For example, 6-mercaptopurine, okay, this is the structure of the 6-mercaptopurine, suppresses the body's immune response and is therefore useful in protecting the donor grafts. Unfortunately, the drugs tend to be eliminated from the body too quickly. The prodrug azathioprine, this is the prodrug azathioprine, okay, it has an advantage that is slowly converted to the 6-mercaptopurine by being attacked by the glutathione, allowing the more sustained activity. The rate of the conversion can be altered depending on the electron withdrawing ability of the heterocyclic group. The greater electron withdrawing power, the faster is the breakdown. The nitro group is therefore present to ensure an efficient conversion of the 6-mercaptopurine as it is strongly electron withdrawing on the heterocyclic ring. So as you can see, this is the structure of the azathioprine, which is here is a thiol, which is there. With this, we have a this particular imidazole ring with a nitro substituent. As we know, the nitro substituent is strongly electron withdrawing, and that is a requirement that the I mean the heterocyclic group must have a group which is a strongly electron withdrawing, and also so there is a glutathione which attacks on this position, and then we have this kind of resonating structure which will have this particular, first is the this elimination of this group, and we will have our I mean the active drug 6-mercaptopurine from the prodrug azathioprine. Okay, and here this nitro group is the particular group which ensures that the particular conversion happens facilely, okay, in a facile manner. So here this glutathione, we have discussed it I think in our 6 to 7 class, I think, when we were discussing about the drug metabolism in the phase 2 metabolism. This thing I think I have discussed a little bit about this glutathione and how it works. Okay, you can read back all the things if you want to more details regarding this phase 2 metabolism. Okay, so remember, nitro group is the group that which is a very electron withdrawing, and it ensures that the 6-mercaptopurine can be it can be derived from the prodrug azathioprine more efficiently. Okay. Now I will go to the next problem. So problem number 5 says which amino acid residue is released from the LDZ, a prodrug of diazepam. Your options are number A valine, number B lysine, number C tyrosine, and number D alanine. Which amino acid residue is released from LDZ, which is a prodrug of diazepam? Your options are valine, lysine, tyrosine, and alanine. I will wait for a minute, then I will check the chat box. Let me know A, B, C, or D, which of the following will be the correct answer for problem number 5. Which amino acid residue is released from LDZ, which is a prodrug of diazepam? Which amino acid residue is released from LDZ, a prodrug for diazepam? Valine, lysine, tyrosine, or alanine, A, B, C, or D, which of the following is the correct answer? Let me check the chat box first. Okay, I have received only one answer which says that number A is the correct answer. Okay, let's find out if number A is the correct answer or not, or is there any different answer? Let me check. Okay. So, for problem number 5, the correct answer, the amino acid residue that is released from LDZ, which is a prodrug of diazepam, will be, it is number B, lysine. Okay, lysine is the amino acid residue which is released from LDZ and it is a prodrug of diazepam. So this is the structure of diazepam. If you look at this structure more, clearly, you will see that there is a lysine residue which lies here. Okay, this is a lysine residue which lies here. So this is the structure of LDZ and this is the structure of diazepam. Okay, so LDZ is an example of diazepam prodrug which avoids the drowsiness side effect associated with the diazepam. These side effects are associated with the high initial plasma levels of diazepam following the administration. The use of the prodrug avoids the problem. An aminopeptidase enzyme hydrolyzes the prodrug to release the non-toxic lysine moiety and the resulting amine spontaneously cyclize to the diazepam. So as you can see, there is a this kind of amide linkage is present here, this particular amino acid lysine is present in this site. So this lysine is cleaved by this aminopeptidase enzyme, and after that this amine is released, which has a lone pair which attacks on this carbonyl carbon, and there is a internal cyclization which creates a 1 2 3 4 5 6 7 member which creates a not 7 8 member ring. Okay. 1, 2, 3, 4, 5, 6, 7. Okay, which creates a particular 7-member ring, and here we will find that there is then internal hydrogen, internal proton, a transfer, and which makes this area position double bond. Okay, so this is the conversion of LDZ to diazepam. If possible, practice this conversion few more time because this mechanism is important. It is very easy and simple to do, but you have to practice this one time. Okay. This LDZ is a very the few are the things that have been kept in mind while designing this particular prodrug. One is the problem, which is the high percentage of diazepam in the bloodstream as it is administered. But if you use the prodrug, the active drug diazepam will be released slowly, so it will not give rise to the high initial plasma level, which is the which is the reason behind this drowsiness associated with this drug. Okay. Second, that the group which is cleaved the lysine is the non-toxic. It is a normal amino acid which is not toxic for our body at all. So these two things which that has been kept in mind while designing this LDZ, which is an the starting prodrug for the diazepam. Okay. So the amino acid which is residue which is released is the lysine. Okay. Now I will go to the next problem. Problem number 6 says, which of the following statements is correct? Your options are number A, Palmitate ester of chloramphenicol increases its water solubility. Number B, palmitate ester of chloramphenicol is given intravenously. Number C, succinate ester of chloramphenicol increases its water solubility. And number D, succinate ester of the chloramphenicol is given orally. Which of the following statement is correct? Again, I will repeat. Number A, palmitate ester of chloramphenicol increases its water solubility. Number B, palmitate ester of chloramphenicol is given intravenously. Number C, succinate ester of chloramphenicol increases its water solubility. And number D, succinate ester of the chloramphenicol is given orally. Which of the following statement is correct? I will give you 1 or 2 minutes, then I will check the chat box. Okay, read the options carefully and then let me know A, B, C or D, which of the following will be the correct answer for problem number 6. Let me check the chat box. Let's see which of the following answers are given in the chat box. Okay. Number C, number A, number A. So I have received two number A and one number C as the answer. Okay, let's find out which of the following will be the correct answer for problem number 6. So the correct answer is number C, succinate ester for chloramphenicol increases its water solubility. So basically, this is the structure of the chloramphenicol when R is the H, okay? And if the the chloramphenicol prodrugs, chloramphenicol palmitate is this group, okay? And chloramphenicol succinate is will have this group as the R. So some drugs have revolting taste. One way to avoid this problem is to reduce their water solubility to prevent them from dissolving in the tongue. For example, bitter taste for the antibiotic chloramphenicol can be avoided by using the palmitate ester. It is more hydrophobic because it is mixed alcohol group with a long chain of the fatty acid group is present here. It does not dissolve easily on the tongue and quickly hydrolyzed once swallowed. Products that have been used to increase water solubility of the drugs, that is the particularly useful for the drugs which are given intravenously, means it has a higher concentration and the small volume can be used. For example, succinate ester of the chloramphenicol increases the latter means the water solubility because it has the extra carboxylic acid group presence. Once the ester is hydrolyzed, chloramphenicol is released along with the succinic acid which is naturally present in our body. So now let us go back to our statement number A, which says palmitate ester increases the water solubility. It is false. Okay, palmitate ester is given intravenously. No, it is given orally to mask the bitter taste. This particular palmitate ester has been used to mask the bitter taste of chloramphenicol so that it cannot get dissolved in the tongue easily. Okay, succinate ester of the chloramphenicol increases the water solubility. Okay, it is correct. And succinate ester is given orally. No, the succinate ester is given intravenously. Okay, and because the succinate esters are, they are released in the chloramphenicol is once the product is hydrolyzed, the chloramphenicol is released, the succinic acid will be released, which is particularly naturally present in our body. So it is an example how the products can use to lower the water solubility and also how the products can be used to improve the water solubility. Means the same drug, chloramphenicol is the one drug, but two different, two different esters can be used or two different esters are used to mark this particular group, okay, of the chloramphenicol, and both are used in a different context. One is used to lower the water solubility and another is used to improve the water solubility. Okay, now I will go to the next problem. Problem number 7 says cyclophosphamide is commonly used in anti-cancer therapy. Which of the following statement is not true? Your options are number A, it is relatively non-toxic. Number B, it acts as a prodrug. Number C, it cannot be taken orally. Number D, the structure is metabolized to release acrolein. Which of the following statement is not true regarding the cyclophosphamide? Again, I will repeat the options. Number A, it is relatively non-toxic. Number B, it acts as a prodrug. Number C, it cannot be taken orally. And number D, the structure is metabolized to release acrolein. Which of the following statement is not true regarding the cyclophosphamide? Regarding cyclophosphamide, how it is used, what is the mechanism of action? I think all of these I have already discussed. I think in our fourth or fifth class when we were discussing about the nucleic acid as drug target, I have discussed about the cyclophosphamide in detail. Okay. Now let me know A, B, C, or D. Which of the following will be the correct answer? Cyclophosphamide is commonly used in anti-cancer therapy. Which of the following statement is not true? Number A, it is relatively non-toxic. Number B, it acts as a prodrug. Number C, it cannot be taken orally. Number D, the structure is metabolized to release acrolein. Which is not true? Read the options carefully and then let me know A, B, C, or D. Which of the following will be the correct answer? Let me wait for a minute and two and then I will check the chat box. Okay. Which of the following statement is not true regarding the cyclophosphamide? Let me check the chat box. Number B, number A, number D. So I think I have received number D, number A, and number B, three different options as answer. Okay, let me check which of the following will be the correct answer, number D, A, or B. So the statement which is not true will be, it is number C. It cannot be taken orally. This statement is false. Okay, cyclophosphamide can be taken orally actually. So this is the structure of the cyclophosphamide. Cyclophosphamide is a successful non-toxic prodrug which can be safely taken orally. Let me highlight this part. Okay, so it is a non-toxic prodrug which can be safely taken orally. Okay. Once absorbed, it is metabolized into the liver into a toxic alkylating agent which is useful in the treatment of cancer. And regarding what is the alkylating agent, what is the mechanism of actions for the alkylating agent, all these I have discussed in detail in our I think it was the fourth class, I hope, that had this nucleic acid as the drug target. There I have discussed the mechanism of action of alkylating agent, particularly about the cyclophosphamide in detail. So this is the structure of the cyclophosphamide. It has okay. So in the after taking it orally, it is metabolized by the cytochrome P450 enzymes to the 4-hydroxyl cyclophosphamide. When there we have a hydroxyl group here. After that, it is converted to the aldophosphamide, and in this phase detoxification can also happen. So this aldophosphamide is converted to the phosphorite muster, which is the active alkylating agent, which is actually this particular this alkylating agent, or it is the toxic for the particular our cancer cell, okay, because it is acts as a alkylating agent and causes the DNA or protein alkylation of the malignant cells or tumor cells, okay. And also there is a acrolein which is released. This acrolein is a I mean it is actually the toxic side product for the which is developed from or which is derived from this cyclophosphamide. But there is a the co-administration of this particular drug with another drug which actually diminishes the toxicity of acrolein, helps to prevents this issue of the toxicity of acrolein, okay. And this is also I have discussed in that particular slide. You can go back to that particular slide and you can read in more detail about the cyclophosphamide, how it works, what is the mechanism of action, and so on, okay. So the correct answer was number C, which says that it cannot be taken orally, but it can safely be taken orally, okay. So now I will go to the next problem. Problem number 8 says, which of the following is the correct reason for using hetacillin as a prodrug of ampicillin? Your options are number A, increasing water solubility, number B, decreasing water solubility, number C, eliminating toxic metabolites, and number D, increasing chemical stability. Which of the following is the correct reason for using hetacillin as the prodrug of ampicillin? Your options are number A, increasing water solubility, number B, decreasing water solubility, number C, eliminating toxic metabolites, and number D, increasing chemical stability. Which of the following is the correct reason for using hetacillin as the prodrug for ampicillin? Which of the following will be the correct reason of using hetacillin? Let me know in the chat box, A, B, C or D. I will wait for 2 minutes, then I will check the chat box, okay? A, B, C or D, what is the reason, correct reason for using hetacillin as the prodrug for ampicillin? Number A, increasing water solubility, number B, decreasing water solubility, number C, eliminating toxic metabolite, and number D, increasing chemical stability. Which of the following is the correct reason for using hetacillin as the prodrug of ampicillin? Which of the following is the correct reason for using hetacillin as the prodrug of ampicillin? Let me now check your answers. Okay. Okay, I think there is no answer for that particular question. Let me show you what is the correct answer and then let us discuss it, okay? So the correct reason or the which is for using hetacillin as the prodrug of ampicillin will be number D, increasing chemical stability. So the antibacterial drug ampicillin decomposes in concentrated aqueous solution as a result of the intramolecular attack for the side chain of the amino acid group on the lactam ring. Hetacillin is the prodrug which locks up the offending nitrogen in the ring position and prevents its reaction. Once the prodrug has been administered, hetacillin slowly decomposes to release the ampicillin and acetone. In the field of antiviral drug, the cyclopropane carboxylic acid esters and being studied as the potential prodrug of aciclovir in order to prolong the chemical stability. So this is the structure of hetacillin, this is the structure of ampicillin, okay? And this particular ampicillin decomposes in the concentrated aqueous solution because of the intramolecular attack of this amine side chain of the lactone ring, okay? But what happens in hetacillin, this particular amine which is prone to metabolization, it is locked in this position so that it cannot be attacked quickly by this particular it cannot be attacked quickly, okay? And after it slowly releases ampicillin, acetone is formed as a side product which is non-toxic, okay? So which is non-toxic. So because ampicillin is not chemically more that much stable, hetacillin has been designed to increase the chemical stability of ampicillin, okay? So if possible, please remember the structures for ampicillin and hetacillin, whatever I mean however, we will be discussing this about this antibacterial drugs of the ampicillin and all these things in the next class. So then you will understand what is to remember, why it is important to remember the particular structure of this ampicillin and its products. Okay. Now I will go to the next problem. Problem number 9 says molecules which are formed by reducing peptide nature in the lead molecule is known as, your options are number A peptidomimetic, number B oligonucleotide, number C prototype and number D neurotransmitter. Molecules which are formed by reducing the peptide nature in the lead molecule is known as number A peptidomimetic, number B oligonucleotide, number C prototype and number D neurotransmitter. Molecules which are formed by reducing the peptide nature in the lead molecule is known as which of the following? Peptidomimetic, oligonucleotide, prototype or neurotransmitter. Again, let me wait for some time, then I will check the chat box. Let me know A, B, C or D, which of the following will be the correct answer for problem number 9. Molecules which are formed by reducing the peptide nature in the lead molecule is known as peptidomimetic, oligonucleotide, prototype or neurotransmitter. Let me know A, B, C or D, which of the following will be the correct answer. Molecules which are formed by reducing the peptide nature of the lead molecule is known as which of the following? Peptidomimetic, oligonucleotide, prototype or neurotransmitter. Let me check the chat box now. Okay. Number C is the given answer. Let me check if number C is the correct answer or not. So the correct answer will be It is number A. Okay, peptidomimetic is the molecule which is formed by reducing the peptide nature of the lead molecule. So, peptidomimetics are small protein-like compounds designed to mimic the 3D structure and biological activity of the natural peptides while overcoming their inherent pharmaceutical flaws. They serve as a bridge between high specificity biologics like proteins and antibodies and the stable small molecule drugs. Why use peptidomimetic? Natural peptides are often poor drug candidate because they are rapidly broken down into by enzymes, have short half-lives and struggle to cross the cell membranes. Peptidomimetics solve these issues by enhancing the stability by replacing natural bonds of the isostars with isostars that enzyme cannot recognize or break. Okay, improving bioavailability by adjusting the solubility and lipophilicity to allow the oral administration instead of the injection. Increasing selectivity, restricting a peptide's shape so it fits only its intended target, reducing the off-target side effects. So these are the reason why peptidomimetics are used and why it is very important to use the peptidomimetic. Common types and strategies, backbone modification. Number one, peptoids. Side chains are shifted from alpha carbon to nitrogen atom, making it more highly resistant to proteases. Beta peptides, and extra carbon is added to the backbone, creating unique helical structure that nature cannot degrade. As a peptide, replacing the backbone carbon with nitrogen atom to fix the molecule into the specific turns. Structural constraints, staple peptide at chemical staple hydrocarbon bridge locks the peptide into an alpha helix shape, helping it penetrate the cells and bind more tightly to the targets. Cyclization, connecting the ends of the side chain of the peptide into the ring like the drug cyclosporine to freeze the active shape and prevent degradation. Non-peptide mimetic, these look nothing like peptides but place key chemical group to the exact same 3D spots as a peptide would, often resulting in a classic small molecule drugs like morphine which mimics the endorphin. Real world example, nirmatrelvir Paxlovid. It is a peptidomimetic that inhibits the SARS-CoV main protease to the treat COVID-19, and desmopressin, a modified version of the hormone vasopressin, which with much higher stability and selectivity. Okay. So this is about the peptidomimetics. What is the peptidomimetic? Why to use peptidomimetic? And what is the example of the peptidomimetic? Okay. Slowly we will discuss more about the peptidomimetic in our upcoming slides. Okay. So now let me go to the next problem. So problem number 10 says, what sort of therapy involves the administration of the antibody linked to an enzyme such that the enzyme activates the prodrug? Again, I will repeat, what sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? Your options are number A, ADAPT, number B, GDAPT, number C, ADEPT, and number D, GDEPT. What sort of therapy involves administration of an antibody linked to an enzyme such that the enzyme activates the prodrug? Number A, ADAPT, number B, GDAPT, number C, ADEPT, and number D, GDEPT. Let me know A, B, C or D which of the following will be the correct answer for problem number 10. I will wait for a minute or two and then I will check the chat box. Okay. Let me know which of the following is the particular therapy or which therapy involves the administration of antibody links to an enzyme such that the enzyme activates the prodrug. Let me check the chat box. Let's see if there is any answer for our problem number 10. Okay, I have received two answers. One is saying number B, another is saying number C. Let me check which of the following is the correct answer, number B or number C. Okay, and then let us discuss it. So, the correct answer will be, okay, 1 second. So, the correct answer is, it is number C, ADEPT. So now, what is the full form of the ADEPT? It is the antibody directed enzyme prodrug therapy. Okay. So the question was, which therapy involves an administration of an antibody linked to an enzyme so that the enzyme activates the prodrug? The therapy is known as the ADEPT, or antibody directed enzyme prodrug therapy. Okay. So antibody directed enzyme prodrug therapy, or ADEPT, involves two steps. First is the administration of an antibody enzyme complex. The antibody is raised against the tumor selective antigen and is linked to the enzyme such as the bacterial carboxypeptidase. This complex then gets bound to the tumor. Unlike the antibody drug conjugates, the antibody enzyme complex needs to remain attached to the surface of the cell and should not be internalized. A certain period of time needs to elapse to give the complex time to bind to the target cells and for unbound complex to be cleared from the blood supply. A prodrug of the cytotoxic drug is then administered. The prodrug is designed such that it will be stable in the blood supply and can only be cleaved and activated by the enzyme complex to the antibody. This means that the toxic drug is then only produced at the tumor and can be administered at the higher dose, can be administered in higher dosage than the parent drug. CGS 149 is an example of one such prodrug which is activated by the bacterial carboxypeptidase. Let me just highlight it for your so that you can remember. So this is an example. Okay, one CGS 149 is an example of the one such product which is activated by the bacterial carboxypeptidase. An advantage of the ADEPT over antibody drug conjugate is that the enzyme is catalytic and can generate a large number of active drug molecules at the site of the tumor. This can then diffuse into the tumor and affect the cells which might not have any antibody attached to them. A lot of research has been carried out on ADEPT using bacterial enzymes such as the carboxypeptidase G2, penicillin G acylase, and beta lactamase. The advantage of using foreign enzyme is that the enzyme can be chosen and that are not present in the mammalian cell, so there is no chance of product being activated by the mammalian enzyme during the circulation around the body. This is very important. Okay, why this bacterial enzymes are used instead of mammalian enzyme? The reason that these enzymes, so that it cannot be activated by the enzyme present in the mammal cell. Okay, so there is no chance of the product being activated by the mammalian enzyme during the circulation around the body. So this is very important thing. Okay. It is also possible to use foreign enzymes which have counterparts in the body as long as the latter are only present in the low levels of the blood and are not they are structurally distinct. Products can be designed to react selectively with the foreign enzyme rather than the mammalian version. Example of the enzymes of the category are the glucuronidase and nitroreductase. Okay, so many studies has been carried out on ADAPT. One example is the antibody beta lactamase complex capable of reacting of the cephalosporin prodrug of an alkylating agent. This takes advantage of the mechanism by the cephalosporin react with beta lactamase to eliminate the leaving group. Okay, so this is the active functional group. This is the prodrug which is used with the active functional group, and this is the adapt which is the antibody linked with this enzyme. Okay, so beta lactamase, it will cleave this react with this particular beta lactam ring of the prodrug, and the active alkylating agent will remove. So this is the adapt strategy to release the alkylating agent. Okay, so is it clear or do I need to repeat it one more time? So basically, this is not very much tough. Okay, once I share the PPT with you, you can go through that particular slide. Okay, and if you do not understand what is ADAPT and why it is used and how it is used actually, so you can let me know via email. I will, or you can let me know via the in the next class also. Before starting the session, I will repeat it one more time. Okay. I hope it is clear. It is very easy. Once you read it by yourself, I think you will clearly understand why, what is ADAPT, why it is used, and what is the role. Okay. So simple things are that first this particular the thing is that the antibody must be linked to the enzyme which is not present in our body, so that the prodrug which is intended to be activated, that particular adapt strategy means by this particular enzyme which is linked with the antibody, it is not present in our body, so that the particular group, the prodrug, it is released, it should release the active drug in the target site itself. Okay. And the example of the adept is also given. So if you are in our final examination, if you are said to write a particular short note, you can use this particular thing and draft a short note regarding the adept therapy, right? So now I will go to the next problem. The problem number 11 says, what is the main challenge for GDEPT? Your options are: number A, designing the active gene; number B, delivering the gene selectively to the tumor cell; number C, identifying the correct enzyme; and number D, all of the above. What is the main challenge for GDEPT? Number A, designing the active gene; number B, delivering the gene selectively to the tumor cell; number C, identifying the correct enzyme; and number D, all of the above. What is the main challenge for GDEPT? I will wait for some time, then I will check the chat box. Okay, after that I will discuss in detail what is GDEPT. So like ADEPT, it is also a different therapy. Okay, and which is also very important in the medicinal chemistry. Let me wait for 1 to 2 minutes, then I will check the chat box. Okay, you think and let me know which of the following will be the main challenge for the GDEPT. Number A, designing the active gene; number B, delivering the gene selectively to the tumor cell; number C, identifying the correct enzyme; and number D, all of the above. What is the main challenge for GDEPT? Number A, designing the active gene; number B, delivering the gene selectively to the tumor cell; number C, identifying the correct enzyme; and number D, all of the above. Let me check the chat box. Okay, there is no answer. Okay, it's fine. Let me show you the correct answer and let us discuss about the GDEPT. So the main challenge for GDPT is number B, delivering the gene selectively to the tumor cell. Now the GDPT is the gene directed enzyme product therapy. Then in the DPT it was the antibody directed enzyme product therapy. It is gene directed enzyme product therapy is the GDPT. So the gene directed enzyme product therapy involves the delivery of the gene to the cancer cell. Once delivered, the gene codes for an enzyme capable of transforming the prodrug into active drug. As the enzyme will be produced inside the cell, the prodrug is required to enter the cell. The main challenge of the GDPT is the delivering the gene selectively to the tumor cells. In one method, the gene is packaged inside the virus, such as the retrovirus or adenovirus. In the case of the adenovirus, the desired genes could be spliced into the viral DNA such that the virus insert into the host cell DNA on infection. The virus is also modified genetically such that it is no longer virulent and can do no harm to the normal cell. Non-viral vectors have also been tried, such as the cationic lipids and peptides. So far, it has been possible to achieve. It has not been possible to achieve the required selectivity for the cancer cells over the mammalian cells, and so the delivery vector has to be administered directly to the tumor. The enzymes which are are ultimately produced by the introduced gene should not be present in the normal cell, so that the product activation only occurs in the tumor cell. This is also very important. Okay, the enzymes which are ultimately produced by the introduced genes should not be present in the normal healthy cell. Okay, so that the product activation only happens in the normal cell. One advantage of GDAPT over ADAPT is the fact that foreign enzymes could be generated inside the cancer cells and hidden from the immune response. The thymidine kinase enzyme produced by the herpes simplex virus has been studied intensively in the GDPT. This enzyme activates the antiviral drug acyclovir and ganciclovir. All these drugs are post substrate for the mammalian thymidine kinase. Activation will only occur significantly in the tumor cell containing the viral form of the enzyme. Several clinical trials have been carried out using this approach. One problem associated with the GDPT is that it is unlikely that all tumor cells will receive the necessary gene to activate the prodrug. It is therefore important to that the anti-cancer drug somehow transfer between the cells to the tumor, a so-called the bystander effect. This may occur by a variety of means, such as release of the activate drug from the infected cell, direct transfer to the through the intercellular junctions, or by the release of the drug carrying vehicle from the following the cell death. GDPT has been used to introduce genes for the bacterial enzyme nitroreductase or carboxypeptidase G2 into the cancer cell. Prodrugs were administered which are converted to the alkylating agent by the result of the enzymes. One of the problems with the carboxypeptidase G2 is the difficulty of some prodrugs have in crossing in the cell membrane. In order to overcome this problem, the gene was modified such that the resulting enzyme was incorporated into the cell membrane with the active site revealed on the outer side of the cell. Gene therapy aimed for the activating the prodrug irinotecan can be explored to try and improve the process by the urethane is hydrolyzed to the active drug. This could be introduction of the gene encoding the more active carboxypeptidase enzyme in the tumor cell. For example, rabbit liver carboxypeptidase is 100 to 1000 times more efficient than the human form of the enzyme. Okay. So this is all about the GDPT. Like ADPT, it is also very important as a product therapy, and short notes may come in your final examination. Okay, regarding these therapies. So read the therapy, read this particular slide very, I mean, carefully. If you do not understand a particular anything, please let me know. I will repeat it one more time in our upcoming class. Okay, if there is any issue, please let me know beforehand, I mean by via email or so that I can to discuss that particular problem in the beginning of the next class. Okay, now I will go to the next problem. Problem number 12 says identify the full form of ADAPT. Identify the full form of ADAPT. Your options are: number A, antibody directed antigen prodrug therapy; number B, antibody directed absorption prodrug therapy; number C, antibody directed antibody prodrug therapy; and number D, antibody directed enzyme prodrug therapy. Which is of the following is the full form of ADAPT? We have discussed about ADPT, GDPT. Now it is the time to discuss the ADAPT. So let me know which is the following is the full form of ADAPT. Number A, antibody directed antigen product therapy. Number B, antibody directed absorption product therapy. Number C, antibody directed antibody product therapy. And number D, antibody directed enzyme product therapy. Let me know. I will wait for a minute, then I will check the chat box. Okay. Identify the full form of ADAPT. Let me wait for another minute, then I will check the chat box. Okay. Let me check the chat box. Let's see if I have received any answer. Okay, number A. Number A. So I have received number A as the answer in both the. Okay. So I have received number A as the answer. Let me check if number A is the correct answer or not. So the full form of ADAPT will be, it is number D. Okay, antibody-directed enzyme prodrug therapy is the full form of ADAPT. So enzymes are the antibodies which have a catalytic property. It is possible that prodrugs can be designed that it can act as an act as an antigen. Now, these antibodies are activated by the enzyme's catalytic property. Okay, now what is enzyme? I think I have discussed in a class when I was discussing about the antigen antibody. Okay, so this can be done by immunizing a mice with a transition state analog of the reaction that is desired, followed by the isolation of the monoclonal antibody by the hybridization techniques. As the antibody targets the prodrug rather than the antigen on the cancer cell, it fails to target drugs to cancer cells. However, it should be possible to construct the hybrid antibodies where one arm recognizes the antigen on the cancer cell while the other arm recognizes the prodrug and activates it. This approach is still in the early stages, but it has several potential advantages over ADEPT. For example, it should be possible to design catalytic mechanisms that do not occur naturally, allowing highly selective activation of the prodrugs at tumor. It also removes the risks of an immune response due to the foreign enzyme. At present, the catalytic activity of the enzymes are too low to be useful, and much more research has to be carried out before using this particular therapy. Okay. So, the full form for this ADAPT is the enzyme prodrug antibody directed enzyme prodrug therapy. Okay. Now I will go to the next problem. Problem number 13 says, which of the following is a peptidomimetic formation technique? Your options are number A, backbone modification, number B, side chain modification, number C, structural mimetics, and number D, all of the above. Which of the following is a peptidomimetic formation technique? Number A, backbone modification, number C, side chain modification, number D, structural mimetics, and number D, all of the above. I think I have discussed a little bit about peptidomimetic in our today's class itself. So I think you can easily answer this question. Which of the following is the peptidomimetic formation technique? Let me know A, B, C or D. Backbone modification, side chain modification, structural mimetic or all of the above. Which of the following is a peptidomimetic formation technique? Let me check the chat box. Let's see if there is any answer there. Okay. I have received one answer saying that number D is the correct one. Okay. Let me check if number D is the correct answer or not. So, the correct answer will be yes, it is number D, all of the above. The backbone modification, side chain modification, structural mimetics, all are the peptidomimetic formation techniques. Okay. So, peptidomimetic formation techniques involve the structural modifications to natural peptides to improve their stability, bioavailability, and binding affinity. These techniques are generally categorized into three more main approaches. One is the backbone modification, then side chain modification, and structural or secondary structure mimetics. So, backbone modification technique, These methods are alter the fundamental peptide chain to enhance the resistance to the enzymatic degradation. Number one is the amide bond isostar, replacing the peptide bond CONH with the bioisostar like triazole, thioamide, or sulfonamide. In alkylation, substituting the nitrogen atom for the backbone with an alkyl group for example, by N-methylation to reduce the hydrogen bonding and improve the cell permeability. As a peptide, replacing alpha carbon of an amino acid with a nitrogen atom, which restrict the backbone's flexibility and often induces the beta turn conformations, and backbone extension, incorporating additional atoms to the chain such as in beta peptide and extra carbon and oligoureas peptides, shifting the side chain from the alpha carbon to the backbone nitrogen, creating N-substituted glycine oligomer that are highly resistant to proteases. Then we have the side chain modification technique. These techniques modify the functional group attached with the peptide backbone to optimize the biological activity. For example, using the unnatural amino acid, incorporating a non-proteogenic amino acid to introduce unique chemical properties or steric bulk. Then side chain substitution using click chemistry and cross metathesis to attach the functional moiety such as sugar and heterocyclic rings directly to the side chain. Then stereochemical alteration substituting L-amino acid with D-enantiomer to significantly increase the metabolic half-life. Structural and secondary structure mimetics. The strategies use global restriction to lock the peptide into the bioactive conformation. For example, macrocyclization, forming cyclic structure like head to tail or side chain to reduce the conformation flexibility and improve oral activity. Now, peptide stapling using hydrocarbon bridges via ring closing metathesis to stabilize the alpha helical structure and enhance the cell penetration. Then hydrogen bond surrogates, thus replacing natural internal hydrogen bonds with covalent linkage to nucleate and stabilize the helical form. And scaffold-based mimetic using small molecule template like ternals and to project the side chain functional group and pattern to mimic the helices. Okay. So these are the techniques by which the peptidomimetics can be formed easily. Okay. Now I will go to the next problem. So problem number 14 says a dash drug is the drug that is given alongside another drug to enhance the latter's activity. Fill in the blanks. Number one, multi. Number B, abzyme. Number C, sentry. And number D, none of the above. A dash drug is a drug that is given alongside another drug to enhance the latter's activity. What will be this drug? Your options are number A, multi drug. Number B, abzyme drug. Number C, sentry drug. And number D, none of the above. I will wait for a minute or I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C or D, which of the following is the correct answer for problem number 14. A dash drug is a drug that is given alongside another drug to enhance the latter's activity, which is the correct answer. I will wait for 2 minutes, then I will check the chat box. Let me know A, B, C or D, which of the following will be the correct answer for problem number 14. Which drug is given alongside another drug to enhance the latter's activity? Let me check if there is any answer for our problem number 14. Okay, number B. I have received one answer which saying that number B is the correct answer. Let me check if number B is the correct answer for problem number 14. So, the drug which is given alongside another drug to enhance the letter's activity is known as the sentry drug. Okay, number C is the correct answer. Sentry drug is the drug that is used alongside the another drug to enhance its activity. So, sentry drug in sentry drug approach, a second drug is administered with the principal drug in order to guard or assist it. Okay, so usually the second drug inhibits the enzyme that metabolizes the principal drug. For example, clavulanic acid inhibits the enzyme beta-lactamase and is therefore able to protect the penicillin from the particular enzyme. Okay, these things I will also discuss in detail in our upcoming class when we will discuss about the antibacterial agent. Okay, so remember the names. Okay, the clavulanic acid, beta-lactamase, and penicillins. The antiviral preparation Caletra used in the treatment of the AIDS in combination with true drugs called the ritonavir and lopinavir. Although the former has an antiviral activity, it is principally present to protect lopinavir, which is metabolized by the metabolic cytochrome P450 enzyme. Ritonavir is a strong inhibitor for this enzyme, and so the metabolism of lopinavir is decreased, allowing the lower doses to be used in the therapeutic plasma level. Another example is found to in the drug therapy is the of the Parkinson disease. The use of levodopa as a prodrug of dopamine has already been discussed. I have also discussed it in our in some class when we were discussing about the drug metabolism. I think there I have discussed about the use of levodopa as a prodrug of the dopamine. Okay, so this is the levodopa and dopamine. The conversion happens with the enzyme dopa decarboxylase. Okay, to be effective, however, large doses of so the use of levodopa to be effective, large doses of levodopa like 3 to 8 gram per day is required, and over a time the dose leads to the side effects such as nausea and vomiting. Levodopa is susceptible to the enzyme dopa decarboxylase, and as a result much of the levodopa administered is decarboxylated to dopamine before reaches to CNS. This buildup of the dopamine in the peripheral blood supply leads to the observed nausea and vomiting. The drug carbidopa is then used as a sentry drug, and has been successfully used as the inhibitor of the dopa decarboxylase enzyme, okay, which allows smaller doses of levodopa to be used. Furthermore, it is also highly polar compound containing two phenolic OH group. As you can see, there are two phenolic OH group. Okay, a hydrazine moiety and a H2 moiety and an acidic group also present in here, and so it is unable to cross the blood-brain barrier. So it cannot prevent the conversion of levodopa to dopamine in the brain. So this is very important. So technically, levodopa is given so that it can release the dopamine inside our brain, right? But what happened, like because of this enzyme dopa decarboxylase, much of the levodopa is already converted to dopamine before it can cross the blood-brain barrier and reach to the brain. So carbidopa is added as a sentry drug, so to inhibit this particular enzyme outside the brain so that the levodopa can remain intact and cross the blood-brain barrier where it can be converted to the dopamine. Okay, so, but this particular carbidopa is highly polar, so it cannot cross blood-brain barrier because we know that particular main polar compound cannot cross the blood-brain barrier. Okay, so the carbidopa is marked as a mixture of levodopa and it's called the co-carbidopa. Several important peptides and proteins could be used as drug if it were not for the fact that they are quickly broken down by protease enzyme. One way around this problem is to inhibit the protease enzymes. Candoxastril is a protease inhibitor which has some potential to the respect of the under clinical evaluation. Finally, the action of the penicillin can be prolonged if they are administered along with the probenecide. Okay. So these are the various examples of the sentry drugs where two drugs are used. One is the principal drug and another drug is to use to protect it from the external degradation, or it is used to enhance its action. Okay. So remember all the examples of the sentry drugs for whatever we have used in this particular question, like clavulanic acid with the penicillin. Okay. Then the Caletra, the preparation of Caletra with the ritonavir and lopanavir, then the use of this particular carbidopa, the drug carbidopa, which has been used as the sentry drug with the levodopa, and the mechanism of action. Okay, so all these are very important. So read it carefully. If you have any doubt or any query, please let me know. I am saying that repeatedly, that if you have any doubt or any query regarding any question, let me know before the next class. I will repeat that particular point. Okay. Let me go to the next problem. Problem number 15 says, which of the following is not an example of peptide drug? Your options are number A, the cyclosporine, number B, enfuvirtide, number C, teriparatide, and number D, artemisinin. Which of the following is not an example of the peptide drug? Cyclosporine, enfuvirtide, teriparatide, or artemisinin? Which of the following will not be an example of peptide drug? Let me know A, B, C, or D. Which of the following will be the correct answer? I will wait for a minute or two, then I will check the chat box, okay? Which of the following is not an example of peptide drug? Let me check the chat box. Let's see. What is the answer that I have received? Okay, number D I have received as the answer. Let me check if number D is the correct answer or not. So the drug which is not an example of peptide drug is, yes, very good. Number D, artemisinin is not an example of peptide drug. This is the structure of cyclosporine. This is the structure of enfuvirtide. This is the structure of teriparatide. This is the structure of artemisinin. From the structure, it itself is clear that all these three are the peptide drugs, but not are the artemisinin. The structures are very, this is not possible to memorize the structures, that you don't need to do it. Okay, but remember the names. So as there are often reluctance to use the peptide as a drug because there are so many pharmacokinetic difficulties that can be encountered while using peptide as a drug. But it does not mean that peptide drugs have no role to play in the medicinal chemistry. For example, immunoresponsive cyclosporine can be administered orally. An important peptide drug, goserelin, which is administered as the subcutaneous implant and is used against the breast and prostate cancer, earning $700 million per year for its maker. Now, in 2003, enfuvirtide was approved for the first new class anti-HIV drugs. It is a polypeptide of 36 amino acid which is injected subcutaneously and offers another weapon to combination therapies used against the HIV. Teriparatide is used in the which is administered by the subcutaneous injection. Peptide drugs can be useful if one chooses right disease and right method of administration. So as you can see, because of their huge structure, they won't be very much water soluble and they cannot be taken orally. So the mode of administration is very important while using the peptide drug. And so these three, cyclosporine, enfuvirtide, and teriparatide, all are examples of the peptide drug, but not the artemisinin. Artemisinin is an example of the anti-malarial drug. Okay. Now I will go to the next problem. Problem number 16 says megestrol acetate has a short duration of activity since it is metabolized at position number 6. This is the position. Placing the methyl group at 6th position increases the duration of action. What is the role of this methyl group to play? Your options are: number A, it is susceptible to metabolic, it is a susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformational blocker. Number D, it is an electron donating group. Again, I will repeat, megestrol acetate has a short duration of activity since it is metabolized in this 6th position. Placing a methyl group in the 6th position will increase the duration of action. What role does the methyl group play? Now, your options are: number A, it is a susceptible metabolic group. Number B, it is a metabolic blocker. Number C, it is a conformation blocker. And number D, it is an electron donating group. Let me know A, B, C or D, which of the following is the correct answer. For problem number 16, again I will wait for a minute, then I will show you the correct answer. What is the role of the methyl group if it is introduced into the 6th position of megestrol acetate? Let me know A, B, C or D. Which of the following is the correct answer? Let me check the chat box now. Okay, I have received one answer saying number C is the correct one. Okay, let me check if number C is the correct one or not. So, yeah, the correct answer will be number B. The methyl group act as a metabolic blocker. Okay, so some drugs are metabolized with the are metabolized by the introduction of the polar group at the particular position in the skeleton. For example, steroid can be oxidized at the position 6 of the tetracyclic ring to introduce a polar hydroxyl group. The introduction of this group allows the formation of polar conjugate, which can be eliminated quickly from the system by introducing the methyl group in this 6th position. So, if include so, by increasing the methyl group at the 6th position of metabolism is blocked and the activity of steroids is prolonged. The oral contraceptive megestrol acetate is an agent which contains 6-methyl blocking group. Okay, so it acts as a metabolic blocker when the group is 6. Okay, and if we want to make this molecule more metabolically susceptible, we would have introduced a more polar group like hydroxyl group. Now, on the same line, a popular method of protecting aromatic rings of the metabolism is a para position to introduce a fluorosubstituent. For example, CGP is an enzyme inhibitor which acts on the kinase effective active site of the epidermal growth factor receptor. It went forward in clinical trial as an this is the example. Okay, this went forward to the clinical trial as an anti-cancer agent and was found to undergo oxidative metabolism in the para position of the aromatic ring. Fluorosubstituent was successfully added to the analog CGP53353 to block the metabolism. This tactic was applied successfully in the design of the gefitinib. Fluorine has now been used extensively to block metabolism in the variety of structural situations. Another approach, which is actively being explored in that to replace the hydrogen with the deuterium isotope. The covalent bond of the carbon and deuterium is twice as strong as the between carbon and hydrogen, and this might help to block the metabolic mechanism. So this is the structure of this particular CGP 52411 when the X is hydrogen, and now when X is equal to fluorine, it is CGP 53353, which uses the fluorine in the para position to block this metabolically susceptible positions. Okay. Now I will go to the next problem. Problem number 17 says, which of the following statement regarding neurotransmitter is not true? Your options are number A, they are secreted by the neuronal cell. Number B, they are chemical messengers. Number C, they basically traverse through blood to act on the distant target cell. Number D, they carry out a cascade of reactions. Which of the following statements regarding neurotransmitters is not true? Number A, they are secreted by the neuronal cell. Number B, they are chemical messenger. Number C, they basically traverse through blood to act on the distant target cell. And number D, they carry out a cascade of reaction. Let me know A, B, C or D, which of the following will be the correct answer. Which of the following statements regarding neurotransmitter is not true? After a minute, I will check the chat box. Let me know A, B, C or D, which of the following you think is the correct answer. Which of the following statements regarding neurotransmitter is not true? Let me check. Okay, I have received one answer saying number D is the correct answer. Let me check if number D is the correct answer or not. So the correct answer is number C. They basically traverse through blood to act on the distance target cell. This statement is not true. Okay. So now let us discuss about the neurotransmitters. So neurotransmitters are body's chemical messengers that allow neurons to communicate between each other as well as with muscles and glands. They traverse across the tiny gap called synapse and to deliver the signals that regulate everything from your heartbeat to your mood. How they work? So basically, release when an electrical impulse reaches to the end of the neuron, it triggers the release of the neurotransmitter stored in the small sacs called vesicles, binding the chemicals across the synapse to bind to the receptors to the target cell, similar to the lock and key mechanism. Depending on the type of neurotransmitter and receptor, the signal will either excite, trigger an action, inhibit, stop an action, or modulate. Fine-tune the target cell response. Removal. To prevent overstimulation, chemicals are cleared away through the reuptake, absorption back to the neuron, degradation, breakdown to by enzyme, or diffusion, drifting away. Key neurotransmitter and their function. Dopamine linked to pleasure, reward, motivation, and motor control imbalances are associated with the Parkinson disease and schizophrenia. Serotonin regulates mood, sleep, appetite, and digestion. It is primary target for antidepressant drugs. Okay, glutamate, the mood abundant excitatory neurotransmitter essential for learning and memory. GABA, the gamma aminobutyric acid is the main inhibitory neurotransmitter. It calms the brain and critical for managing anxiety and preventing seizures. Acetylcholine controls muscle contraction, plays a major role in memory. Low level are linked to the Alzheimer disease. Norepinephrine or epinephrine key players on fight or flight, stress hormone response, increasing heart rate and alertness. Okay, so these are the key neurotransmitter and their function. So basically, they do not traverse through the blood distance cell to act on the distant cell. They actually act near it is secreted. Okay. Now let me go to the next problem. Problem number 18 says, which of the following statement is incorrect? Your options are: number A, ticonazole can only be applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole is better interaction with blood. Number D, ticonazole can only be applied intravenously. Which of the following statement is incorrect? Again, I will repeat. Number A, ticonazole can only be applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole has better interaction with blood. And number D, ticonazole can only be applied intravenously. Which of the following statement is incorrect? Read all the options carefully and then let me know A, B, C or D, which of the following will be the correct answer. Which of the following statements is incorrect? Number A, ticonazole can be only applied through skin. Number B, fluconazole is more polar than ticonazole. Number C, fluconazole has better interaction with blood. And number D, ticonazole can only be applied intravenously. Which of the following statements is incorrect? Let me know. Which of the following statements is incorrect? Let me check the chat box first. Okay. Number D. Let me check if number D is the correct answer or not. So correct answer is very good. It is number D. Ticonazole can only be applied intravenously. This statement is false. So this is the structure of ticonazole. This is the structure of fluconazole. So basically, adding or removing a polar functional group to vary the polarity of the drug, a polar functional group could be added to the drug to increase its polarity. For example, the antifungal drug ticonazole can only be used for skin infection because it is a non-polar and poorly soluble in blood. Introducing a polar hydroxyl group is a more polar heterocyclic rings lead orally active antifungal agent fluconazole with improved stability and enhanced activity against the systematic infection that is in the blood supply. So this is the structure of the ticonazole, which do not have many polar functional groups. So it is very much insoluble in blood, and also it can be only used topically, means through the for skin infection. Okay, but fluconazole is another antifungal drug, and as you can see, there are hydroxyl group, there are triazole ring, there are fluorine, which makes this molecule very much polar. So this particular antifungal drug can be used orally Okay, and because it is orally active and it is can be used against the systematic infection like the infection which happens in our systematic circulation in blood, that can be tackled by the fluconazole. But ticonazole can only tackle the infection that happens on topically on the skin, because it is a very much non-polar. Okay. Now I will go to the next problem. Problem number 19 says there are several strategies for stabilizing susceptible bonds in the peptide. Which of the following is not used in this respect? Your options are: number A, replace a peptide link to the stable functional group. Number B, adding N-methyl group to the peptide link. Number C, using unnatural amino acid. Number D, replacing D amino acid with L amino acid. Which of the following strategy is not used for stabilizing susceptible bonds in peptide? Your options are: number A, replacing a peptide link with stable functional group. Number B, adding N-methyl group to the peptide link. Number C, using an unnatural amino acid. And number D, replacing D amino acid with L amino acid. Let me know A, B, C or D which of the following is the correct answer for problem number 19. I will wait for a minute, then I will check the chat box. Okay. Which of the following is the correct answer for problem number 19? Let me check if there is any answer in the chat box. Okay, there is no answer for this question. Okay, let me show you the correct answer and let us discuss it. Okay. I think a part of this I have already discussed when I was discussing the peptidomimetic, right? So the strategy which is not used for stabilizing the susceptible bond in peptide will be number D, replacing D amino acid with L amino acid. If you remember, now when I was discussing the strategies to make the peptidomimetic, I have discussed that replacing a peptide link with stable functional group or by various amide isosters is the method. Then adding N-methyl group and using unnatural amino acids are another methods for introducing the peptidomimetic, but not the D amino acid to the replacement of replacing D amino acid with L amino acid. It is not that much important. Okay, what is the differences between the D and L amino acid? By the way, so the L amino acid is stereoisomer of the particular amino acid whose amino group is on the left side of the Fisher position, and D amino acid is a stereoisomer where the amino group of the right side of the Fisher projection. L amino acid can rotate the plane polarized light counterclockwise in a process called levorotation. D amino acid can rotate the plane polarized light clockwise in a process called dextrorotation. L amino acid suppressed by the R notation and superseded by R notation, and L amino acid D amino acid is superseded by S notation. L amino acid used by cell to produce protein, whereas the D amino acid occur in the cell wall of bacteria. Okay, so these are the differences between L and D amino acid. Now, the last question of today's class: why morpholine and pyridine rings are added to the drug molecules? Your options are: number A, to improve hydrophobicity, number B, to improve water solubility, number C, to increase the molecular weight, and number D, all of the above. Why morpholine and pyridine ring are added to the drug molecule? Your options are: number A, to improve hydrophobicity, number B, to improve water solubility, number C, to increase the molecular weight, and number D, all of the above. Let me know A, B, C or D which of the following will be the correct answer for problem number 20. So last question of today's class. Okay. Why morpholine and pyridine rings are added to the drug molecules? To improve hydrophobicity, to improve water solubility, to increase molecular weight, or all of the above? Why morphine and pyridine rings are added to the drug molecules? What is the correct reason? Yeah, let me check the chat box. Okay, I have received one answer which says that number B is the correct one. Let me check if number B is the correct answer or not. So the correct answer is yes, number B, to improve the water solubility. This is the structure of morpholine. This is the structure of pyridine. Okay, so the first question to explain why morpholine and in many times what we have find that drug molecules is actually quite poorly soluble. So in order to improve the solubility, one needs to add groups which can be hydrophilic in nature, but they are not going to dramatically alter the properties of the drug. So here the nitrogen containing heterocyclics such as the morpholine, this one, and pyridine are quite often used, so that the nitrogen is the polar and definitely increases the polarity in some cases. The protonated form can also be isolated as water soluble salt. So here is an example. If you connect through the nitrogen, then you can sort the whole molecule. Morpholine is quite polar, and so here in case of pyridine, you can make pyridinium salt actually isolated from the pyridinium salt. So if the polar group is added in order to increase the solubility, it is preferable to be added to the molecule in such a way that it is still exposed surrounding the water. Okay, so therefore, because it is exposed to surrounding water, the drug is still bound to the target site, does not have to sort to dissolve it before binding to the active site. So it is important criterion that we are trying to add more functional group to the drug. Okay. So the basic reason why this morpholine and pyridine are added to drug molecule is that to increase this water solubility. So this was the last question of today's session. So today in today's class, basically we have majorly discussed about the prodrug, various therapies which are related to the prodrugs like ADPT, GDPT, ADAPT. We have seen lots of examples of the prodrugs, and also we have discussed regarding the peptidomimetic, the methods by which peptidomimetics can be formed, and what is the use of this, what that we have discussed in today's class. So now I will share my email ID. If you have any doubt or any query regarding today's class or any of our previous classes, you can let me know via this email. Okay. Now, I will surely repeat that particular topic, whatever you find it is difficult in the upcoming classes. Okay. And today it was our now I think live session number 9. It is our ninth class. We will have three more regular classes. Okay, three more regular classes we will be having in our course along with the summary class that regarding the schedule that will be communicated to you later on. Okay, so where to find the material? I have already shown you a couple of times that you have to go to the medicinal chemistry, the page, the SWAM portal. On the left panel, you have to click on the problem solving session recording. So you will be redirected to a page which looks like this. This will contain the particular recording link, and also it will contain a Google Drive link which will contain the presentation which I am showing in the class. Okay. So if you have any doubt or any query, you can let me know. Otherwise, I will close the session for today. And we will meet on our next class, which will be on next Monday from 6 p.m. The PPT and the class recording I will upload. I will try to upload it by tonight. Okay. So you can check and you can go through the materials. If you find any difficulty understanding any topic, please let me know. I will discuss it in detail in our upcoming class. Okay. So is there any problem? Is there any question or doubt? Otherwise, I will close the session for today. Okay, thank you all for joining. Please let me know if you have any problem via email, or you can drop your query in the discussion forum also. I will try to answer your queries in the upcoming classes. Okay. I will upload the materials so you can check it later tonight or tomorrow. You can check it on the SWAM portal. Thank you all. I am closing the session for today. Let's meet on the next Monday. Okay. Thank you.